morning, everyone. Thank you for coming. My name is Michael Clem. I'm an associate in JPM's Healthcare and Investment Banking Group. It's my pleasure to introduce our next speaker, Ms. Sarah Boyce, the President and CEO of Avidity Biosciences.
Thank you. At Avidity, we are creating a new class of RNA therapeutics using our proprietary antibody oligonucleotide conjugate technology. What that technology enables is what we believe to be an answer to one of the fundamental challenges that has impacted the delivery of RNA therapeutics, which is, can you effectively deliver therapy to dense tissues like muscle, heart, and also be able to target cells like B cells, T cells, to open up the immunology space and the immuno-oncology space. What I'm going to share today is data from our AOC platform that leads us to believe that we've been able to address that fundamental. We've been able to give the answer to that fundamental question. I'm Sarah Boyce, I'm the CEO of Avidity, and I'd like to thank the JPM team for giving me the opportunity to present the company to you today. Our mission is creation, development, and delivery. And when I say creation, the AOC platform technology was created at Avidity. So it is our proprietary platform designed to overcome the delivery limitations of oligonucleotides. And what we've been able to do is demonstrate modulation of disease in many of RNAs in many different tissue types, including muscle, heart, liver, tumor, and immune cells. Our strategy is based around developing our internal programs of our rare muscle portfolio, and I'll present that pipeline to you today, as well as also partnering with pharma to fully realize the potential of our therapeutic platform. I'll also talk today about our partnership with Lilly, that is an example of that. The fundamental goal is to advance our science to the clinic as rapidly as possible to be able to provide effective therapies for patients. So what is an AOC therapeutic? We can use a different monoclonal antibody to target different types of cells and different tissues. We attach an oligonucleotide to that antibody. So the antibody is the targeting mechanism to be able to have the oligonucleotide access the cell, and the oligonucleotide is the therapeutic. We're often asked what matters. Is it the antibody? Is it how you bind the oligonucleotide to the antibody? Is it the oligonucleotide itself? Is it where you bind on the monoclonal antibody? The simple answer is absolutely everything mattered in the development of this technology, both looking at monoclonal antibody selection, looking to binding locations, how we bind the oligonucleotide to the monoclonal antibody, and of course the oligonucleotide itself. Just taking a little deeper look on the oligonucleotide, one of our other advantages is that we're not actually limited by type of oligonucleotide therapy. What you'll see is in our muscle pipeline, where our platform is siRNA, but in particular, when we're targeting DMD, for example, so when we're targeting a splicing disorder, we're utilizing a PMO. So what we're able to achieve is a very potent, durable mechanism of action, and I'll show you the data around that, as well as realizing the potential for pretty infrequent dosing. And we're looking at dosing potentially of once a quarter. And we're able to deliver our therapies to previously undruggable targets for RNA therapeutics. From a pipeline perspective, you'll see our rare muscle programs. Our program in myotonic dystrophy is our lead program that we're looking to advance into the clinic next year. That's closely followed by our muscle atrophy program. We're actually taking a completely different approach from others who have targeted muscle atrophy. Previously, one of the goals of muscle atrophy is to build muscle. What we're able to do by using an siRNA is be highly selective as the type of target that we look at. And in this case, targeting MUF1, which is a gene expressed in atrophying muscle. So essentially, we're looking to try and stop the atrophy. We also have a program in Duchenne's muscular dystrophy. Here we show programs in exon 44 and 45, but we're also able to target we're also able to target many of the relevant exons in DMD. We also have a program in FSHD, as well as in Pompe disease. Our pipeline outside of muscle is growing, and in part through our collaboration with Lilly. We're very proud to have Lilly as a partner, and through our partnership with Lilly, we've been able to advance our immunology program. It is a partnership that is restricted to a set number of targets. So in parallel in working with Lilly, we can also develop our own immunology programs as well. So looking at our focus and how you'll see us expand into a broad range of therapeutic areas, our immediate focus is our lead program in myotonic dystrophy. From the near term, then expanding out into other rare muscle disorders, be DMD, FHSD, and Pompe disease, and then in the longer term, expanding through the platform to look at multiple therapeutic areas, including heart, immunology, immuno-oncology as examples. It's through this broad potential that we were able to recently complete our Series C round. We raised over $100 million, and that round was led by RTW. We're very proud to have such a prestigious group of investors partnering with us at Avidity. I lead a very experienced team. The experience spans from large pharma as well as also to starting companies. And that broad range of experience we've proven to be highly useful from an aspect of anticipating where we need to scale up earlier in our efforts, for example in manufacturing, in terms of really getting in front of that. I'm partnered in particular with Art Levin, who is our Chief Scientific Officer. Art is a leader in this field, in the oligonucleotide field. We joke that Art has in many cases forgotten more than most people actually will ever know in this field. And there truly is no question. You can ask that gentleman and he doesn't know the answer. So we have a fantastic team that I'm very proud to lead. Going in more to our platform and our technology. So AOCs are designed, as I said, to fundamentally improve on RNA drug properties. Today, there are 5 commercially available RNA drugs. I personally led the launch of 2 of those drugs and was closely involved in a third. So this is a field that I know very well. RNA therapeutics have been transformative. Through RNA therapeutics, we've already seen diseases that were previously untreatable be able to be addressed. There's a high potency and you can be very specific. With AOCs, we're looking to build on that in bringing the properties of RNA therapeutics, but also through our AOC technology, being able to provide a far superior PK/PD versus unconjugated drugs, as well as being able to get that activity outside of non-hepatic tissues. This is an example of our preclinical work on a proof of concept. This is looking, so some of these are different controls for different cell types. One of the things that you will see, which I think no one will be surprised, is being able to achieve a very robust target reduction in the liver. But what's really important is the other tissues and the other cell types on the rest of this slide, because this is where there have been fundamental challenges with RNA therapeutics, and this is where we've been able to show great activity in our models, in particular in muscle, in being able to achieve a 95% reduction in our target RNA. That leads us into the development of our rare muscle pipeline and our rare muscle disease programs. Our lead program is in myotonic dystrophy. Myotonic dystrophy is a devastating rare disease. It's estimated to be about 130,000 people impacted with myotonic dystrophy in the US today. Not all of them, of course, are diagnosed. Given a rare disease, you have some of the classic challenges. It is a monogenic disease, but it is a multi-system disorder. It primarily affects skeletal and cardiac muscle. The cause of death in 70% of these patients is cardiac and pulmonary. It has a devastating impact on their quality of life. Over time, there is a progressive weakness in a range of muscles in the face, so people lose that expressive ability, which actually impacts social interaction, but then also in arms and legs. One of the hallmarks of the disease is myotonia, so this is where the muscle is slow to release. So what it gives us from a clinical trial perspective is a great marker. So if you look at grip, what happens with patients with myotonic dystrophy, they can grip, but that grip release time is very slow. And that's something that we can measure in clinical trials. Now, when you think of that release of a contracted muscle, it's not just about the grip strength and an inability to release a handshake. Think of that impact on your leg muscles, for example, if you're not able to release the contraction of those muscles. And that's where you get an inability and restrictions from being able to walk and ultimate mobility restrictions. Myotonic dystrophy is an ideal disease and target for us with our AOC therapies. It's caused by a single mutated gene, which is the DMPK gene, and that gene leads to multiple splicing errors in muscle messenger RNA. So what happens is these people with myotonic dystrophy have CUG repeats. Now, you and I probably have in our DMPK gene somewhere between 5 to 37 repeats. These patients can have thousands. What happens with these CUG repeats is they start to bind on each other, and as they bind on each other, you'll start to get hairpins and hairpin loops form. Those loops attract muscle-blind-like messenger RNA, and that gets sequestered within the nucleus. One of the things that then happens is that leads to multiple downstream splicing errors in a whole range of different muscle-related messenger RNAs. So single cause resulting in a cascade effect. So if we can knock down the DMPK messenger RNA expression, then we can have an impact on that cascade effect on the splicing proteins. There is a validation to this hypothesis through a clinical trial conducted with an unconjugated oligonucleotide, where there was the ability to show some of that movement, but that program isn't in development for the challenges around targeting muscle. And this is where we come in with our AOC technology. Looking at an unconjugated AOC, that was looking at a micromolar concentration in the cells. We're able to achieve nanomolar of a potency through the siRNA mechanism. That is a threefold order of magnitude difference over what can be achieved with an unconjugated oligonucleotide, achieving with a single dose with a long duration of action, potentially over months. So looking at the data. So this is looking at a range of different leg muscles and looking at DMPK expression reduction in those models. This is at 3 different doses, 1.5, 2.5, and 5 milligrams. That 1.5 dose is at an ED50, and it is an equivalent to a nanomolar concentration. This type of robust knockdown in muscle tissue is something that has us extremely excited for a potential coming into the clinic. Now, really importantly, is looking at that over time. So what we're showing here is single dose, 1 injection, 5 milligrams per kg, and expression over 6 weeks. In the same types of muscles, again, you see that continued knockdown over time. Importantly, we then went to look at 2 different types of tissue, heart and diaphragm. You remember earlier I said why these are so important as being one of the causes of mortality in these patients. And again, we were able to see a very robust knockdown in our target in these tissues as well. So from an overall clinical profile, we see reduction in target messenger RNA in the relevant disease models. We're looking to target improvement in biomarkers and able to do that with a very potent approach and a long duration of action. We're right now establishing we've established on manufacturing and analytics, and are moving towards the clinic and expecting to start our first in-human trial next year. This is just a glimpse of one of our other muscle programs, and this is D&D. We also know in D&D that, again, effective delivery of oligonucleotides to the muscle has been a real problem. And this is a comparison to an unconjugated PMO versus a PMO conjugated to our antibodies. We were able to achieve a 50-fold increase in exon skipping. In actual fact, for the first time, we were able to saturate that. So we're really excited to also be advancing forward our DMD program into the clinic as rapidly as possible. Now, taking a brief look outside of our muscle programs and into another types of cells, looking at immunology and immuno-oncology. This data is both in vitro and in vivo, looking at producing specific expression in B cells and T cells. What similar pattern as to what you've been seeing before, again, effective knockdown of our target through our AOC technology. It was this data in particular that had Lilly very excited and got their attention, and was the foundation of our partnership with Lilly. The Lilly partnership involved an upfront payment and an investment in our Series C. It is a multi-targeted program, and there is a specific number of targets in which we're working on with them, but it also, importantly, allows us to develop our own targets on our own program in immunology and immuno-oncology ourselves, as well as also working together with Lilly. And they're a great partner, and we're really excited as to how we've been able to advance the technology much faster than we otherwise would have been able to do on our own through this partnership with Lilly. So in terms of looking at how we believe we're pioneering a new class of targeted RNA therapeutics, which is our AOC platform, our strategy is to develop our internal muscle programs, looking to partner with pharma to really, truly realize the full potential of the platform. Our partnership with Lilly is an example of that, and bringing our science to previously undruggable tissues and cells and messenger RNA targets to provide therapies ultimately for patients who otherwise wouldn't have had it. From an upcoming catalyst, one of our key elements is marching towards the clinic and advancing our lead program in myotonic dystrophy to the clinic, planning to initiate those studies next year, and as well as also advancing the rest of our pipeline in muscle space and our partnership with Lilly. We believe at Avidity that one of the things we have been able to do and have the promise to do is to answer one of the fundamental challenges around RNA therapeutics, which has been effective delivery to muscle and to a whole range of other cells. I've shown you a brief aspect of our data in our program, but I hope I've given you a sense as to why we believe that's possible.